Mycobacteria tuberculosis have distinguished into three classes. The number one is a photochromogen, number two sclerochromogen, and non-chromogen. First, we will explain the photochromogen. And photochromogen is a non-tuberculosis Mycobacteria. The M. kinsasi. The epidemiology of M. kinsasi is usually found common in white males. The natural reservoir is tape water. Aerosol is the way its transmission. The type of infection is chronic pulmonary diseases. M. asiaticum is not commonly encountered. It's mostly found in Australia, and potentially pathogenic in pulmonary disease is the type of infection asiaticum cause. M. merinum is a natural reservoir in the fresh water, salt water. As a result, a contamination from the infected fish or marine life. Transmission by contact with the contaminated water or organism entirely by the trauma or small break in the skin associated with the aquatic activities usually involving this fish, and the infection caused by merinum is a bacteremia or a cutaneous disease. Mycobacterium intermedium is known potentially pathogenic organism, and pulmonary disease is the type of the infection M. intermedium cause. M. novaca strain epidemiology is unknown, just like intermedium, and potentially pathogenic and cause the cutaneous diseases. Photochromogen is again differentiated on the basis of biochemical activities. These are a slow growing non-tuberculosis mycobacteria. If we see the nitrate test is the primary test, which differentiated further by tolerate test from M. sulexi. M. sulexi is basically photochromogen only at 25 degrees centigrade. Usually it is classified in sclerochromogen because it do not require the light to show the pigmentation. M. kinsasi is tolerate negative, and further asiaticum is nitrate negative, urease negative, and also in this group merinum is twin hydrolysis positive, and semi is twin hydrolysis negative. And photochromogen, the merinum is nicene positive and yellow to dark orange color colony. Pigmentations are produced mostly found in the swimming pools, water, skin's complication, as we explained. M. gordoni is a tape water bacilli. It's another rapid grower which is mentioned here. Non-tuberculosis mycobacteria classified as sclerochromogen. M. sulexi, as we explained over there. This is water found in epidemiology is in water in the soil, and potentially pathogenic organism cause pulmonary preeminently in the middle-aged man, cervical adenitis, and bronchitis. And this M. scrophilicum is a raw milk or soil in water or dairy product, potentially pathogenic cervical adenitis in the children, bacteremia, pulmonary diseases, skin infection. M. interjectum is have unknown epidemiology, potentially pathogenic chronic lymphadenitis, pulmonary diseases. M. hexhumans is potentially pathogenic and pulmonary diseases. M. tussi is also isolated from the tape water, potentially pathogenic cervical lymphadenitis. Only one case is reported so far. M. cubici is unknown epidemiology, potentially pathogenic and pulmonary diseases. M. gordoni, M. cookie, and M. hebemi is tape water, surface water in the New Zealand, and in Ireland hebeni is found, and all of three are non-pathogenic. In non-photochromogen, the two new species M. Chelatum 25 and N. conspicum 26 is also appear to be potentially pathogenic in humans. These are newly identified species in non-photochromogen. In scrotochromogen, non-photochromogen, here in this chart the identification is followed by nitrate, which is further negative, and followed by NaCl. And if NaCl is negative, nitrate is positive, that is Sulagzi, which is 25 on 25 degrees centigrade, pulmonary diseases, and TB in middle age patient. M. flavescens is positive for nitrate and also for NaCl, and that can be identified as rapid grower also. Scrotochromogen, if the nitrate is negative and the urea is also negative, then we go for the test to check the temperature tolerance, which is on 45 degrees centigrade. If it is positive, then it is M. zunopi. M. zunopi is a bird nest like colony from the back of the plate, isolated from the African toad Xenopus. It's mostly involved in the diabetes patient and malignancy or alcoholism. If nitrate is negative and urea is positive, then we go, the suspected organism is Mycobacterium scrophilicum, that is required 3 to 4 weeks in the children. It causes cervical lymphadenitis. It's also have urease positive. In scrotochromogyl, if nitrate is negative and urease is negative and also organism is unable to grow on 45 degrees centigrade, then we consider that organism is M. gordonii. It's rapid, rarely cases of the human isolate and tape water bacilli, and tolerate reduction test is negative for this organism. Rapid grower non-tuberculosis mycobacteria have 8 groups that are pathogenic and weak gram-positive rods resembling dipterioids, Chinese letter. M. fortutum group, M. cheloni, M. abscessus is commonly encountered. These 3 species account for approximately 90% of a clinical